So NEOSTEM is a public company. I am going to be making some forward-looking statements, and most of you in the room know the significance of this slide. So let me start by congratulating Dr. Van Bockelen for his introductory section a little while ago, where he highlighted the probably single greatest problem facing developed nations around the globe, and that is the ever-increasing cost of treating chronic disease. And the paradigm, as he articulated, I agree with completely, and that is that if we don't address this with transformative therapies, that will change the paradigm from active treatment of a chronic disease to one in which, where you make it as if you didn't have the disease at all, we are going to be in trouble. My background is I'm a practicing bone marrow transplant physician. I've been an experimental therapeutics development physician for over 30 years. I was involved in hematopoietic growth factor development, monoclonal antibody development, and a number of cancer drugs. And what I'd like to do as I present AMR 001 is highlight some of the features in the spirit that Dr. Van Bockelen started the session with in regard to the things that you really need to focus on if we're going to be successful as a field into developing experimental therapeutics. So the first is in cell therapies. The first is that cell therapies, no matter how they're manufactured and delivered, they're not going to be inexpensive. And they are going to need to address a large unmet need. So I'll start with acute myocardial infarction. I know my colleagues will be discussing other aspects of cardiovascular disease. The incidence of AMI is decreasing, and the severity of AMIs have been decreasing over the last 15 to 20 years. But nonetheless, in the most recent data from the NHLBI, there's approximately 800,000 AMIs in the U.S., 20% of which represents 160,000 Americans, and in Europe that number is about 150,000. They have a severe enough MI, they're called STEMIs, where they're at risk to experience progressive deterioration in heart muscle function, leading to premature death, recurrent myocardial infarction, and congestive heart failure. So the question is, can you prevent that? Well, in order to prevent something, you have to understand why it occurs. And why it occurs, at least in this setting, is a consequence of inadequate perfusion, microvascular insufficiency, leading to hibernating cardiomyocytes and progressive cardiomyocyte loss due to apoptosis. And in this cartoon here, you can see, so the infarct area here, that's dead, and our particular therapy does not address that. But let's say you lose 20% of your myocardium. It's a zero-sum game because you don't really generate new cardiomyocytes all that rapidly. So the remaining 80% have to work at 1.2 times their current workload, but it's not linearly distributed. So the cells in the peri-infarct zone are under incredible stress, and many of those cells undergo salvage mechanisms and pathways where they go into hibernation, and if they stay in hibernation too long, they die through apoptosis. So your infarct, which was a 20% infarct 6 months later, is a 25% infarct, and on, and that's why you have these events. So understanding that and looking at cell therapies, if you're going to take a cell from a person and give it back, there has to be a rationale why you're taking that cell and what does it do, and Jason alluded to that in his list of things. And this is critically important for investors and for people in development to really understand why a particular cell in a given indication. So it turns out after an AMI, there is a natural response to that. Every cell in the body, when it's exposed to hypoxia, has hypoxia-inducible factor 1 alpha and beta, translocate from the cytoplasm into the nucleus, and it turns on the hypoxia response. There's about 200 genes that turn on. And one of the most important set of genes results in the production of something called SDF1. And SDF1 gets released by the hypoxic cardiomyocytes, not the dead tissue, into the bloodstream, and it calls for cells from the bone marrow that express the CD34 antigen and the CXCR4 receptor. And these cells can actually be measured after an AMI. They increase in circulating amount, and there have been papers published in the New England Journal of Medicine showing a highly significant correlation between circulating frequency of these cells and the extent to which the MI is mitigated. So the greater number of cells in the circulation, the less size of the MI you have a year later, and the better your outcomes. Understanding that and other things, and knowing through animal models that we have done and others, that when these cells are mobilized to the peri-infarct zone, they prevent apoptosis through paracrine factors, and in the interest of time, I won't get into the details, but we do have the details, and affect neoangiogenesis. Again, I can share that later. So we realized that this cell population was the target cell population, and there is over 1,000 patient experience worldwide using unmanipulated bone marrow in this setting with a range of clinical effectiveness, including significant clinical benefit in some studies. But the variability was hard to explain. We now understand why there was variability. One, most of the cells in bone marrow have nothing to do with the effect. It's these cells. And two, these cells have to have a certain biologic property in order to be effective, which I will address in a second. So AMR-001, our lead candidate product, is an autologous bone marrow derived, and I'll spend a moment on pharmaceutical grade, therapeutic intended to preserve heart muscle function and limit the adverse consequences, so-called MACE, major adverse cardiac events, and other adverse clinical events following an AMI. It's pharmaceutical grade because we spent the proper time, as others are doing now, defining identity, potency, purity, relevant biologic stability. What's the shelf life of your product? How do you know that 24, 48, 72 hours later your product is still effective? You have to understand, well, what does it take to be effective? And in our case, in this circumstance, the response, the ability of the cells to be mobile in an SDF1 gradient is what needs to persist. Sterility, dose threshold, and we demonstrated all of this in our phase 1 trial, which I'll walk you through quickly. You have to understand your mechanism of action. How do you know that you have enough cells? How do you know that the cells are the right cells? How do you know the cells, when they arrive in the cardiac cath suite and they've been there for 48 hours, they're still any good? So you have to understand these things in order to answer those questions. And in our case, in this particular circumstance, it's based on SDF1 mobility. You have to have a dominant IP position. We have both composition of matter and methods patents, and I'm going to describe this a little bit more. And this morning we made an announcement about a very significant expansion of our patent portfolio. Manufacturing distribution costs that will allow for commercial margins. We worked on this from the very beginning to make certain that if we ever were fortunate enough to have an FDA-approved product, that we can deliver a product that would allow for proper commercial margins, as Jason expressed. We already have in place existing manufacturing capacity available for the first 2 years of commercialization through Progenitor Cell Therapy, one of our sister organizations within Neostem. And also, you have to demonstrate in this new healthcare environment pharmacoeconomic benefit, which we plan to do. So let me very rapidly go through the phase 1 trial. This was a phase 1 trial done several years ago in patients who had an AMI, and it turns out that if you wait 4 days after a heart attack and measure ejection fraction, you can assess an at-risk group that is in the group of patients that have a high likelihood of having bad outcomes. It's sort of a poor man's biomarker, if you will, to select a subgroup of STEMI patients that are at risk. What we did was we measured perfusion because our basis for the mechanism of action is that CD34 cells expressing a CXCR4 receptor were trophic to the peri-infarct zone, those hypoxic cardiomyocytes, preventing them from dying, and they do that by inhibiting apoptosis and increasing perfusion. So we had to prove that. What was the effect on ejection fraction? What was the effect on cardiac remodeling? Did you prevent a drop in ejection fraction? Did you preserve muscle function? And did this concept of SDF1 mobility play out? And so what we did was we took a patient population that was at high risk. We gave them a single dose. This was a classic dose escalation trial. The first cohort received 5 million cells against a control group, second 10 million, third 15 million. We picked these numbers of CD34 cells based on extrapolation from the unmanipulated bone marrow data, knowing that somewhere in here would be the sweet spot. It was 31 patients at 4 sites. And the first and most important slide I'm going to show you is that we in fact found a highly significant correlation with a p-value of 0.1 between the dose of not just CD34 cells, but CD34 cells that were mobile. Remember, in order for the cells to have their effect, they have to transmigrate through the endothelial wall, because these cells are given via the coronary artery, get to the peri-infarct zone. If they don't get there, they're not going to work. We had expected this, but in the phase 1 trial, we demonstrated it that both in regard to decrease in infarct size measured by CMR and increase in perfusion, that the more mobile CD34 cells you gave, the greater the effect. And that's in fact why we got composition of matter patents. Next, we looked at, okay, but you can't isolate cells by the CXCR4 receptor, because if you place a monoclonal antibody on it, you actually blind the cell. It's not mobile. So you have to do it by isolating CD34 cells. Not all CD34 cells express the CXCR4 receptor. Not all CD34 cells, patient to patient, have the same mobility. So how do you quelch all this biological mobility? As a bone marrow transplant doctor, we figured this out 20 years ago in bone marrow transplant, and the same applies, and I suspect it's going to apply to all cell therapy. Everyone has to get used to the concept of a dose threshold. It's not going to be a linear dose effect with cells. What it'll be is that you're not going to see any effect, or in very few patients you'll see an effect until you get to a first level. Once you get to that level, you start to see an effect. And there may be second or third dose thresholds. But in this trial and with AMR-001, once 10 million cells were administered, in the 5 million group, there was no improvement in perfusion. In the control group, there was no improvement in perfusion. Our TSS score is a hypoperfusion score, so it has to go down for there to be an increase in perfusion. That's just the way the test is measured. And in the 10 million group and the 15 million group, highly significant increase in perfusion. Well, did that translate to an effect on cardiac function? Because of this was highly significant, P01. Because of small numbers and variability, even with CMR, we didn't hit significance, but it came very close in that those patients above the threshold had an absolute increase of 4.5% in ejection fraction, and those below the threshold had no effect. Those above the threshold in regard to ventricular remodeling and systolic volume, you want it to go down over time. They went down versus those below the threshold, they went up. But here's the most striking part of this paper that was published by Dr. Kiwami in the American Heart Journal and presented at the AHA meeting a year ago. In the control group, 30% of the patients had a drop in their ejection fraction, a significant drop 6 months later. In the 5 million group, that was a subtherapeutic dose of cells, 40%. None of the patients, not a single one in the 10 or 15 million group, had a drop in their ejection fraction. So in this trial, albeit small, and absolutely needs to be confirmed in the trial I'm about to present to you, we saw that if you increase perfusion, you prevent adverse remodeling, and you preserve heart muscle function. So we have now launched a phase 2 trial. It is actively accruing patients. It just opened within the past several weeks. This is it's called the Preserve AMI, Preservation of Heart Function. Post-AMI, Preservation of Cardiac Function. The primary endpoint is to, again, demonstrate increase in perfusion. The secondary endpoint, and we're powered for all of these things, is to assess overall cardiac function, including preservation of ejection fraction and adverse remodeling. And we're going to be measuring every clinical event one can think of, quality of life, MACE, and other events. This is a single-dose trial. All patients will receive cell dose above the threshold. Here again, comments this morning, we found the FDA incredibly cooperative and collaborative with us after deeply diving into our phase 1 data, coming up with the concept of dose for the phase 2 trial. This is a randomized 1-to-1 treatment sham placebo control, 160 patients. All patients will have bone marrow harvested. All patients will have something infused into their coronary artery. The control group gets the media that the cells are in. The treatment group gets the cells. 34 sites, U.S. We expect to accrue this year. 6 months from the end of this year, we will have the first look at data. And very briefly, patients come in, have the standard of care after they have a STEMI. You have to wait 96 hours to make certain that you are getting the at-risk group, because many patients have low ejection fractions a day or two after their heart attack. But by 4 days, if you're not better, you're in trouble. And we use CMR as the measure. Patients then undergo bone marrow harvest. We isolate and purify the cells via patented technology, and the cells are administered via a single intracoronary artery infusion. In summary, we have taken a concept and a product and have developed it down a drug pathway with purity, identity, potency. We're working on a biomarker as we speak. And in addition, as a consequence of the way we developed this drug, and I would encourage other companies to do the same, our initial patent portfolio that was granted was for cardiac post-MI versus early and late. As of the 3rd of this year, we got a very significant expansion that any tissue with post-ischemic injury, we have composition of matter now and methods patents as well. And this will allow us to move into congestive heart failure, which we plan to do this year. We have an NHLBI-approved trial that we'll be launching later this year, chronic myocardial ischemia, critical ischemia. And we've also worked out some more practical issues where you can cryopreserve for future use of our product. So let me stop here and thank you very much.